Good morning. It's August 6th, and this is your daily brief in science. Here's everything you need to know. A new study has unveiled the potential of using ultrasound waves to activate brain circuits in living animals, marking a significant advancement in neuroscience. Conducted by researchers from NYU Langone Health and the University of Zurich, the study utilized a helmet equipped with 512 ultrasound emitters to generate holographic patterns that precisely directed sound waves onto targeted regions of the brain. Published in Nature Biomedical Engineering, the findings indicate that entire neural networks in a living mouse brain can be activated by focusing on circuits spread across different brain areas. Co-senior author Shai Shoham highlighted that this transcranial ultrasound stimulation, or TUS technique, can enhance neuron sensitivity to ultrasound by up to 10-fold, which may improve treatment efficiency and safety. This method stands in contrast to current FDA-approved treatments for conditions like Parkinson's disease, which often involve damaging neurons. Instead, this innovative approach temporarily activates neurons without inflicting harm. Achieving effective ultrasound therapy necessitates precise targeting and careful calibration of wave strength to prevent damage to brain tissue. Future research is set to delve into more complex neural circuits and explore deeper brain activation possibilities using ultrasound, with some applications already undergoing clinical testing. The study, which received funding from the National Institutes of Health and the Swiss National Science Foundation, involved a collaborative team of researchers, including postdoctoral fellow Theo Lemaire. The research introduces a novel system that integrates ultrasound sources with a fiber scope to visualize brain activation in mice, potentially paving the way for advancements in treatments for neurological diseases and mental health disorders. Co-senior authors Shoham and Daniel Rozansky noted that the TUS technique could reduce the power needed for ultrasound stimulation, enhancing both safety and efficiency in future applications. NASA's Curiosity rover has recently marked a significant milestone, celebrating its 13th anniversary on Mars. This achievement highlights over a decade of exploration and scientific discovery on the red planet. Engineers have made notable enhancements to Curiosity's productivity, increasing its autonomy and multitasking capabilities, which allows the rover to perform tasks more efficiently than ever before. Unlike its solar-powered predecessors, Curiosity operates using a multi-mission radioisotope thermoelectric generator, or MMRTG, which generates energy from decaying plutonium. This continuous power supply is crucial for sustaining its long-term mission, especially as the energy output diminishes over time. To optimize energy usage, Curiosity has begun combining tasks, such as transmitting data to an orbiter while conducting other activities, which helps reduce overall power consumption. Recent advancements have also enabled Curiosity to autonomously decide when to take breaks after completing tasks early, conserving energy for future operations. Currently, the rover is exploring an area with boxwork formations on Mount Sharp, which may provide insights into Mars's ancient climate and the potential for past microbial life. Equipped with various scientific instruments and cameras, efficient power management is critical for Curiosity's operations. Engineers at NASA's Jet Propulsion Laboratory continuously adapt Curiosity's power budget to meet the challenges posed by the Martian environment, including dust and temperature fluctuations. Over the years, software updates and mechanical adjustments have improved Curiosity's drilling capabilities and reduced wear on its wheels. Having traveled 22 miles on Mars, Curiosity is expected to remain operational for years to come, continuing its valuable scientific research as part of NASA's broader Mars exploration program. A recent study published in The Lancet Rheumatology has revealed that cognitive functional therapy, or CFT, offers long-term relief for chronic lower back pain, a condition that affects 1 in 5 adults in the United States. Developed by researchers at Macquarie University and Curtin University in Australia, CFT is a tailored psychotherapy that has demonstrated effectiveness in relieving chronic lower back pain for up to 3 years following just 8 sessions. The RESTORE randomized controlled trial involved 492 patients suffering from chronic low back pain. Participants were divided into groups receiving either CFT, CFT with biofeedback, or usual care. The findings indicate that CFT can lead to significant improvements, even in patients who have endured disabling back pain for years and have previously tried multiple treatments without success. In a follow-up conducted 3 years later, participants who underwent CFT maintained nearly a 3-fold improvement in pain and disability compared to those receiving usual care. Dr. Rohan Jotwani expressed optimism about CFT's potential for lasting relief, noting that it is uncommon for patients to experience such extended benefits from a single intervention. Lead author Dr. Mark Hancock emphasized that CFT adopts a holistic approach, empowering patients to manage their pain while addressing various relevant factors. The therapy aims to shift patients' mindsets about pain, moving away from fear-driven behaviors and fostering self-management skills that persist beyond therapy sessions. CFT sessions include assessments of posture, pain perceptions, emotions, and lifestyle, helping participants alter pain-related behaviors and adopt healthier habits. However, the study also highlights the need for scaling up clinician training to enhance accessibility and implementation of CFT across diverse healthcare systems. Dr. Jotwani pointed out the importance of addressing psychological barriers, such as kinesiophobia, which can exacerbate pain by limiting movement. Despite the promising results, researchers noted limitations, including dropout rates and the study's exclusively Australian participant base, raising questions about the therapy's applicability in other populations. A recent study has uncovered a key player in the fight against drug-resistant infections. Led by Assistant Professor Ryoji Miyazaki from the Nara Institute of Science and Technology, the research focuses on the small protein LPTM, which is vital for the stability and maturation of the LPTDE complex in gram-negative bacteria. The outer membrane of these bacteria acts as a protective barrier, contributing to their antibiotic resistance and virulence. Understanding this membrane is crucial for developing strategies to combat drug-resistant infections, making this study particularly significant. The LPT transport system is essential for constructing this protective layer, especially through the assembly of the LPTDE complex, which is made up of LPTD and LPTE. While the components of the LPT pathway have been identified, the specific mechanisms behind their assembly and maturation were not well understood until now. Using advanced techniques such as cryo-electron microscopy, researchers revealed the structure of the LPT-DEM complex and demonstrated how LPTM affects the already folded intermediates of LPTD during its maturation. The findings, published in the journal Cell Reports, show that LPTM interacts directly with LPTD, which is critical for the assembly of the lipopolysaccharide transport system. This research suggests that LPTM fine-tunes LPTD's structure, an important factor for LPT assembly and future antibiotic development. A specific region of LPTM, consisting of fewer than 10 amino acids, was identified as essential for its function, highlighting the often overlooked role of small proteins in larger membrane protein complex assembly. The implications of this study extend to potential advances in antibiotic design, as targeting the LPTDE complex could lead to novel antibiotic therapies. Overall, the research emphasizes the importance of understanding the assembly and maintenance of the outer membrane in the ongoing battle against drug-resistant infections. Researchers at UT Southwestern Medical Center have made a significant finding regarding the production of ribosomes during a key stage of neurodevelopment. Led by Dr. Michael Buschak, the team sought to understand the implications of a decline in ribosome levels during early brain development, particularly during neuroepithelial differentiation. This reduction in ribosomes makes developing brain cells particularly vulnerable to changes in ribosome production. Using organoid models, the researchers observed that mutated cells formed smaller brain organoids with higher cell death rates compared to non-mutated cells by day 15 of development. Both organoid types exhibited decreased ribosome production, but those with AIRIM mutations displayed an even greater reduction, resulting in lower levels of essential proteins necessary for cell survival and differentiation. In a breakthrough, the team enhanced mTOR activity, a protein that promotes ribosome production, successfully rescuing the mutated cells and allowing them to develop similarly to non-mutated cells. Dr. Buschak indicated that targeting ribosome deficiencies could lead to interventions for certain neurodevelopmental disorders before birth, potentially alleviating symptoms linked to these genetic conditions. The findings, published in Nature Cell Biology, underscore the critical role of ribosome biogenesis in neurodevelopmental disorders associated with severe intellectual disabilities and sensory impairments due to mutations in the AIRIM gene. The researchers plan to explore whether similar declines in ribosome levels occur in other neurodevelopmental disorders caused by genetic mutations affecting ribosome-related genes. Overall, this research highlights the importance of ribosome production in early neurodevelopment and its potential implications for understanding various neurodevelopmental disorders. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.